everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 3, Episode 5 of Surfing the NASH Tsunami. Our replay of a very special conversation between Scott Friedman and Donna Cryer starts now.
This week on Surfing the NASH Tsunami.
I'm straddling two worlds as a physician-scientist. I'm awed and thrilled by the progress in the science and sobered by the slower progress that's going on in the clinical trial space. There is zero doubt that there will be therapies that treat this disease.
If that agent can do good things fairly quickly, that we might start to think about patient treatment as a pathway. So basically, there's a combination platform that's what am I doing at any given point in time, and then there's a longitudinal combination platform. Some of the ground to think about that way was laid in this meeting.
The early drafts of our patient-focused drug development meeting are available to attendees here at the conference. It was very clear from that meeting, from the patients, that we want something. It doesn't have to be perfect. We understand that there will be iterations and improvements as there have been in every other therapeutic area, but not to have anything at this point is a huge disappointment.
Over the past 10 days, Surfing the NASH Tsunami compiled over 4 hours of recordings from NASH-TAG 2022, available only in episodes running an hour or longer. This week, Surfing NASH is presenting edits from these recordings focusing on specific issues or conversations of interest.
Today, listen to hepatology researcher and key opinion leader, Professor Scott Friedman, Global Liver Institute founder and CEO, Donna Cryer, and our host, pricing and forecasting guru Roger Green, as they discuss NASH and NAFLD from the patient and clinician points of view, followed by a separate conversation between Roger and patient liver wellness advocate and co-host Louise Campbell discussing the first day of NASH-TAG, all on Surfing the NASH Tsunami.
I want to start off today with a kind of a mini business section coupled with a little bit of consumer psychology and perhaps some lessons learned about this podcast. Let me start with the consumer psychology. For this podcast to grow, we need to provide a constant stream of high-quality content. Last year, that led us to cover NASH-TAG by doing a different episode for every half-day session. We produced 290 minutes of high-quality content in 6 episodes, running 40 to 50 minutes each, and we were really excited. But the individual episodes didn't get the kinds of listener numbers we expected, and when we checked in with listeners and downloaders, we found out that by offering so many episodes, we overwhelmed people. Nobody did more than one a day, and they didn't all do the same one every day, on the first day or the second day or the third day. This year, we took a different approach. We decided to do only one episode a day. We did the first business day of the new year, one on each of the 2 meeting days of NASH-TAG, and we did a wrap-up the following Monday, total of 4 episodes. The average episode ran slightly over an hour, much longer than the 45 minutes last year. But by giving people only one thing a day to pick up, the strategy worked great on a listener-downloader level. We measure 7-day listens, 28-day listens, and life of event listens. In terms of 7-day listens and downloads actually, but in terms of 7-day listens, the NASH-TAG episodes of this year are number 3, 4, 6, and 7 out of the 112 original episodes we've done over the life of Surfing NASH Tsunami. Despite the fact that they are somewhere between 8 and 15 days old, on the 28-day list, there are already numbers 8, 9, 10, and 19. These are big numbers. These episodes have done really well. There is one problem, though, making sense of the entire body of work. Our tracking tells us that a decent proportion of people checked out of each podcast within half an hour to 45 minutes, which means you missed a lot of the best stuff at the end. And when I've asked people what they like best about our coverage and what they remember, virtually everyone has mentioned something they heard in the first half hour of whatever podcast they were listening to. So this week, we're going to go back to the NASH-TAG coverage one more time to look for moments that are really special and themes that are really important. Today's episode includes something you really couldn't find anywhere if you didn't listen to Surfing NASH at NASH-TAG 22, a 35-minute conversation between 2 of the most prominent and inspiring people in NASH land today, the father of fibrosis, Professor Scott Friedman from Mount Sinai, and the heroically inspiring Donna Cryer, founder and CEO of the Global Liver Institute. I'm also there because they need an MC, but they're really the guts of the conversation. When that finishes, I've pulled a 10-minute segment that Louise Campbell and I had Saturday morning after our session with Stephen Mazanuruddin and Quentin Anstey, also covering some different topics we don't usually get to. This weekend, we'll have one session discussion looking back at NIT coverage in the aftermath of the fireside chats, a collection of discussions about combination therapy, which will center on the idea that combination therapy for NASH will have not only which drugs are you taking together, but also a longitudinal component. What do you do at what point in the disease progression? And then one more surprise episode that I'll announce on Sunday morning, or I'll announce Saturday for Sunday morning. After this episode, I'll be back with a brief business section to share some listener numbers and let you know about the next 2 weeks of podcasts. For now, though, enjoy Scott and Donna. As the third person in the room, I can tell you the intellect, the passion, and the mutual respect between these two could have fueled a very large boat, or maybe provided enough snow to keep Deer Valley in business all the way through the end of June. Sit back and enjoy it. I'll come back after that.
Donna, do our audience and Scott me a favor and give us one example of something you heard yesterday that will make it easier for you to advocate and make you more effective in doing so. Because we all know no one's more effective than you are when you put your mind to it. So let's do one takeaway from yesterday at least that's going to make it easier to do that well.
My favorite phrase that thankfully I heard multiple times was lessons learned. So the fact that we are not losing the benefit of what has been done and not calling things failures, but really looking at all the hard work that has gone into the field and extracting the lessons from them so that we can move further, faster from this point on.
Okay. Is there one example you can give of a lesson we have learned that makes this helpful?
I think it is as we see it translating into the combinations of drugs and the mechanisms of actions, so that what we learned was that we shouldn't necessarily put all of our eggs in one basket or think that there's going to be one monotherapy or one mechanism of action, one drug to rule them all, but really that it's more likely to be a combination of drugs, a combination of mechanisms of actions to really move the needle to create favorable outcomes for any individual patient.
Okay, super. I want to come back to that because it's one of the things I was fascinated by yesterday. Scott, let's turn to you first. And something that struck you yesterday as being really important.
Well, first of all, I'm honored to share the platform with Donna.
And we're both honored to share it with you. So this is
it's a mutual admiration society, but so be it.
Except I'm the disreputable one here, but go ahead, it's good.
So the way I framed yesterday's sessions were that there was both sobering news, continued unmet needs, but some real promise. The sobering news is mostly on the clinical side. The promise is mostly on the basic and translational science and technology development side. And also I would say there was a major conceptual sea change in my view in that everyone now agrees that digital methods for studies that employ biopsy have to be validated and integrated into clinical trials. So let me back up a little bit and talk a little bit more about the sobering part. There was evidence that despite early data in a number of different therapeutic trials that suggested response, the larger trials did not pan out, FGF19 and FGF21 in particular. And of course that disappoints everybody, patients most of all, but also the providers. And truth be told, I worry a little bit because a lot of the growth is built around investment in small companies, and investors are getting very skittish because they see there are no short-term home runs. I would still say, though, there are many short-term singles, doubles, and even triples. Also sobering is that we don't, as Donna said, have a real grip on where this disease starts, or in other words, I like to say, what's the Achilles heel here? We have, a plethora of abnormalities in fat, inflammation, both within the liver and in other tissues, but we don't know where it starts. Treating one of the abnormalities may or may not be central to the pathogenesis of the disease. And moreover, we tend to still approach the disease as one size fits all. All patients with NASH have exactly the same disease, the same therapeutic targets, and therefore will respond to the same drugs. And I think the analogy that's apt here is to cancer, where for any given cancer we would give the same chemotherapy. Now we look at cancer, even in a single organ, liver in particular, as many different diseases with many different disease drivers and many different causes, and therefore many different therapeutic targets. And so that transitions my comments to the promising side of the world, which is that we saw tremendous progress in many areas that leverage new technology, digital methods for pathology, which I've already alluded to, AI-based approaches. At least 3 different platforms were introduced from 3 different companies. We saw maturation of our understanding in gene regulation and epigenetics epigenetics, that is the non-DNA coding regulatory pathways that change gene expression. We saw continued refinement and engagement in diagnostics. We saw the maturation of organoid technologies and the promise of phage therapies to alter the composition of the microbiome in the gut in patients with NASH. And then finally, we saw stunning data from Matthias Heichenwalder outlining his seminal work identifying the immunologic abnormalities that predispose to NASH liver cancer that are different than liver cancer from other etiologies like hepatitis B and C. So I'm straddling two worlds as a physician scientist. I'm awed and thrilled by the progress in the science and sobered by the slower progress that's going on in the clinical trial space. But to my view, there is zero doubt that there will be therapies that treat this disease. And one of the reasons I say that is because of the natural resolution of the disease in specific patients with NASH. I'm referring mostly to bariatric surgery or sustained weight loss, where we know that the liver harbors this capacity to resorb scar. So clearly the liver knows how to do it under very specific circumstances of weight loss. We need to replicate what the liver already knows and find out what those pathways are and then exploit them using the right therapies. And finally, the disease may be more than one disease, and that comes back to the analogy I gave with cancer, which is that we assume that every patient who has a picture of NASH under the microscope has the same reasons they got there. And that may and probably is not true. And so what we are moving towards, but not quite yet, is identifying different subtypes of NASH that may be responsive to different types of treatments and maybe even different diagnostics.
I should share with you two things that came out of the conversation this morning, one of which you'll find heartening, I think, and the other of which we'll figure out in a minute. We wound up talking about, Stephen was wondering why more people were disappointed about the FGF21 presentation than encouraged. Reason being, they took a look at pegbilfermin and they said, well, at 48 weeks, the entire effect dried up. So how do we know that isn't going to happen with the other agents as well? Where that took us was, if that agent can do good things fairly quickly, that we might start to think about patient treatment as a pathway, not just think about combination therapies, which is what you're doing at one point in time.
Sequential.
But right. So basically, there's a combination platform that's what am I doing at any given point in time, and then there's a longitudinal combination platform. And I think some of the ground to think about that way was laid in this meeting. People are going to have to get there. But I thought that was encouraging. I thought Vincent Wong did a really nice job of explaining, without saying this in so many words, that the aldafermin failure was more commercial failure, really, than it was a clinical failure per se. In one part because it's hard to envision a daily injectable drug being a satisfactory long-term therapy, but in another part because of what the company was dealing with and what other options they had. And Michael made that point in the beginning of your statement about the small companies are doing the development. And the risk there, as you point out, is they might not get the money. The other risk is that they can't afford to cover the whole landscape. So
the final risk is they can't afford more than one failure.
Yeah, well, but by the way, probably some can't afford a success either, right? If you take a look at the trial that Merck is doing with the drug they licensed from aldafermin, it is the trial that we'd like to see lots of the small companies do if they had the money to do it. It's 52 weeks. It's lots of patients. The cells are all well-powered, and there's a logic for everything they did in it. And a lot of the smaller company trials, the companies just don't have the latitude, financial latitude really, to do all that.
Right.
Well, one of the things that de-risks trials is talking to patients early on. Many folks here, have panels of patients advisors on how to develop patient-centered trials, how to keep them as simple as possible while getting the valuable data that they need. And so I think that there is still some room for people to adopt that particular de-risking strategy. One of the very specific cases in which I've had NASH patients deliver insights is on their willingness to take different modes of therapy. So we could answer the question of, is something daily, weekly, oral, injectable? What is the line? What is the trade-off that the people who will ultimately take the drug would satisfy them? And that is information that you could bring back then to your boards or to your investors that said, we've talked to our ultimate customer and they have given us this information, this with feedback. And so rather than simply talking about or hoping for pulling sort of patient centricity from the sky, it's an actual tangible business operating process at this point that can be and should be included in the very early stages, but all along through drug development, to be able to produce a product that best meets the needs in the most efficient way possible.
I think that's half of it, and I don't.
I'm speaking beyond this one example, but
when I say I think that's half of what I mean, I think you're right. Okay, that people can improve their odds by doing that.
The molecule has to work. The molecule is the molecule, but in terms of the
even
design.
so try it this way, right? The landscape is. I'm just going to make this up 6 feet wide, right? If you're big pharma, you can cover 5 of those 6 feet. So if it's there, you'll have a much better chance of finding it. If you're a small company, you can cover 2 of those 6 feet, which means if you're not right in the right place.
But it makes it more important
Wait.
to make sure that you pick the right 2.
That's what I was about to say. You take your bad odds and you improve them by doing what you just described. They're still a lot more exposed than big pharma is, except that big pharma doesn't do this anymore, right? I mean, as Michael pointed out, well, I've been saying this for 30 years. Big pharma has increasingly become like Hollywood studios. Hollywood doesn't make movies anymore. Hollywood buys movies that other people make. And when they decide it's too risky to go with movies that people make creatively, that's when you get Rocky 7 meets Godzilla 4, because we both know those formulas work. It's hard to get big pharma to do what you want them to do. So that's one of the things that, and I think you're right. I think we have to be smart about all the ways that we can play our odds better. But
one other element which Donna knows very well is with respect to patient engagement is the disappointment when things don't work. And in some cases, the lack of a suitable trial. I'm thinking of a woman who's a retired educator who has advanced NASH and has had family history of advanced NASH and complications. So she knows what's potentially ahead of her, and she is, let's just say, committed, if not anxious, to get something that will give her more hope than just waiting for a liver transplant. And that's very painful for me as a physician to hear, and sometimes not to have a trial suitable for her to at least give her hope.
Absolutely. The early drafts of our patient-focused drug development meeting are available to attendees here at the conference. It was very clear from that meeting, from the patients, that we want something. It doesn't have to be perfect. We understand that there will be iterations and improvements, as there have been in every other therapeutic area. But not to have anything at this point is a huge disappointment. But something else that Scott said gives me additional hope, and I hope that everybody picked up on it, was for opportunities for an expanded research agenda. So I think we've been very much in trotting over the same ground in a lot in terms of trial design, and understandably so, given the need to meet 2 particular regulatory endpoints. However, hearing today at this meeting so much more about the potentials for weight loss from surgery, looking at glucose levels and looking at intramuscular fat, and just seeing far more interdisciplinary thinking about how to approach this problem, how to define the problem, and what the path to solutions may be, I think is exciting, both because I think it will ultimately yield fuller answers, but I also think looking at the field of liver health more broadly in the next few years, it gives us so many other ripe avenues for development.
That makes excellent sense. I will tell you that the fastest uptake episode we did in the 4th quarter was Ali Aminian, who nobody knows in this space, talking about Splendor, which is a bariatric study, right? With a really powerful message about cirrhosis and weight loss. And people really, in terms of listening, people really picked up on that. And I think the reason is because it was so hopeful. And from a different place and a different avenue, right? But really a hopeful study.
That's exactly the point I'm making, and it's why I'm hopeful, because we know there's biology underlying this that will get us there. We just don't know which parts of the multi-pronged biology is the one that's really the most vulnerable point to affect an improvement.
The other point you made, I think, is that it'll be different points for different patients, right?
Yes, we think so, or I think so.
It's funny you talk about cancer. The first time I ever showed up here was in NASH 2019. One of the things I talked about was Nixon, 50 years ago then, declaring the war on cancer like there was a drug that was going to cure all cancer, right?
Exactly right.
Right.
That's exactly the right analogy.
And all the things you're talking about, first we found out that different organs were different cancers. And the one I chose was breast, because it was well before liver. We knew that breast was lots of different cancers.
Right.
And then eventually you find some common principles. I think it's hopeful that Scott Harris was talking about about the Ultimmune drug this morning, right? There are 2 or 3 of those dual, and I think Samuel Klein's going to talk about it this afternoon. There are 2 or 3 of those dual or triple agonists that are putting up 20% weight loss numbers in oral agents that you can take daily that don't seem to have particularly troubling side effect profiles. That would be huge if it holds.
The irony is, and I may have said this on our prior podcast, Roger, but if you were to make a grid and list all the effects the ideal NASH drug would develop, the only one that checks all the boxes is weight loss.
Yep.
Other drugs check some of the boxes of what you'd like to accomplish. Other boxes checked by other drugs. The only one that checks all the boxes is weight loss. So what on earth is weight loss doing that's reversing the biology that we can't seem to accomplish with pharmacotherapies?
Or can we, right? I mean, that's going to be the next
ultimately question. We will.
Well, part of the problem with weight loss is it commands a level of human discipline.
Yeah, I mean, we're hardwired to preserve these calories and weight, and largely as we lose weight, and this has been shown in bariatric surgery and other contexts, the body conspires to push us back up to the weight we started at. And Mike Charlton gave the example that's been well advertised in the New York Times and elsewhere, which is the sad outcome of the Biggest Loser participants in that show, which is that they all gained the weight, sometimes more, and when studied metabolically, there was clearly a physiologic conspiracy to get them back to their old obese weight.
Yeah, I was once 50 pounds heavier than I am right now, and it's a struggle.
Very hard.
It's a struggle.
And you're the exception.
Yeah, I'm lucky.
So it speaks to the need for pathways that are multidisciplinary, multifaceted, that don't depend on someone's willpower, but that really recognize all that we know about the psychology and the biology of weight loss and maintenance of weight loss to support people through better use of weight loss medications, better use of diabetes medications, better use of, even by the time we get to a drug that has a specific indication for NASH, we now are, all on record, and I think as Trevor knows, as if you didn't know, now you know, that weight loss has to be a part of this equation. And so how, with even the things that we have now, how can you put together all the pieces of what we like to call care to better enable a person in personalized, as the last great word that I've started to hear, in a personalized way to be successful and to be successful long term. I think that's the question we need to start answering, and it becomes a much more disease agnostic, device agnostic. It's more about the person and the patient than all of these different players. Patients are pragmatic, and so let's figure out what works in what combination, in what sequence, for what profile of person. I think we can create a lot of winners across the field, both on the drug and the device side, and if we really think about how do we continue to drive results for different profiles of patients.
I think that's well said. The idea of personalizing is not just on the medicine side. It's really, I love to listen to Donna because she truly represents the patient perspective. And personalizing is also how the individual copes with the disease, how they make choices, what are the emotional consequences of their illness and or the therapeutic options that are available to them. That's personalized every bit as much as what genes do they express and what risk factors do they harbor.
Oh, yeah. You only have to have one bout of locally advanced cancer in your background to understand all that in a real hurry.
Yeah.
It changes everything.
So let's bring that, all that,
even if you're not a liver patient.
Well,
go ahead.
Let's bring all of that expertise to the liver field. I remember, early on as a transplant recipient, feeling very jealous of cancer patients for all the different things that they were able to receive in terms of a more holistic care. And so while GLI certainly tries to learn the lessons from advocacy from our breast cancer and HIV and hepatitis C colleagues, we absolutely also want to borrow the excellent full elements of 360-degree care that exist for other diseases and bring them to the benefit of liver patients.
That is an amazing comment. that you were envious of cancer patients because of the more holistic approach to their care and dealing with their illness. That is amazing. And I never really thought of it. There is an example that does come to mind that's a very extreme one, which is that when living-related donor transplantation became very hot, everybody focused on the recipient getting their life restored and less so on the donor. And there were some adverse outcomes that shook up the entire field and said, in the donors, and shook up the entire field to say, hey, wait a minute, the donor we owe them as much or more attention, support, programs, both as they work their way up towards donating a piece of their liver, and maybe more importantly, after they've undergone what is major surgery voluntarily.
Yeah.
So,
well, altruistically.
Absolutely.
I mean, it's not just voluntarily, right? It's voluntarily and it's altruistically.
Yes, and
you volunteer to do something because they're going to give you $1 million to do it, or you can volunteer to do it for completely altruistic reasons, which is what makes this so amazing.
Absolutely right. And that's an extreme example, but I hadn't really thought of it in the more broad context in which the way Donna stated it.
So let me give you the flip side, because this came up this morning, right? We were talking about pruritis.
Yes.
And I was told 11 years ago that I needed to get 4 doses of Yervoy into me if I wanted to be sure I was going to live. I had a recurrence of cancer at that point in time. The median survival for recurrence was 15 months. Yervoy was the only solution we could think of. It turns out that my white cells had taken out the tumor, which made it look a little better. And I was in such agony after 2 doses from the level of itching that I was experiencing, and the idea that as somebody who would, part of how I lose the weight and keep it off is I work out like a fiend.
Right.
My blood pressure was 100/60 with 80 beats a minute, and I was falling asleep standing up, sitting in chairs in the middle of meetings. And I just said, I hope 2 doses is enough because I can't do this anymore.
Yeah.
I'm not going to make it another 6 weeks this way. It's going to make me totally nuts.
Yeah.
So a moment when you might not have been jealous to be a cancer patient. But
Well, no, I've experienced pruritis, of course, as a rare liver disease patient, having had cholestatic disease, and it's interesting now that there are medications for pruritis, and I am just going to throw out the question to the field of whether it will continue to be for an ultra rare indication when pruritis is so much a factor across so many different types of liver disease. It sort of stretches my, rather incredulous that it will not ultimately be used across a wider swath of conditions.
Well, one small reassurance, I work with a couple of companies that have helped uncover completely new biology that explains, or that may well explain pruritus far better than the hokey theories that we've been laboring under for the last 40 years. And so there's, as with so many things, there's hope on the horizon.
So let me ask you a different kind of question, though, right?
Yeah.
Because if you go into one of the things that makes oncology special is everyone was, and most still are, cowboys. Labeling means little, right? There's a general faith in drugs, and if I can figure out kind of what this does, I will figure out where I can take it.
Right.
At the lay level, at least, I don't see a lot of that in hepatology. I don't hear about it a lot in hepatology. That same willingness to go out of bounds to the same degree and in the same way. Maybe the top people, but you get one level down from that, and it's not something.
You mean for patients or for
professionals treating patients? How far out of label will I take a drug because I think it might do something?
I can't speak for the practice community, I think, but hepatologists tend to be very conservative, and that's partly it's the temperament or the phenotype of the people that choose it, but it's partly also because many of our patients have slowly progressing disease, often with no symptoms for years or decades, and so they're adhering to the first do no harm admonition of our specialty or of our of medicine, and so trying something outside the norms, number one, is less comfortable for them in an asymptomatic patient, and of course doctors worry about liability too.
Sure. So yes, but I was going back to Donna's comment about why are they using this in an ultra rare condition when so many people have pruritis, and I think that's kind of where you're sitting. Different specialties, you might get a different answer.
But make no mistake. I mean, hepatologists have been tried to be as creative as they can using different approaches to pruritis. Sadly, none of them really work very well. There was naloxone and rifampicin, and of course steroids, and a whole host of and cholestyramine. Donna's probably seen or heard of most of them.
Been on all of them.
So it's not for lack of trying, but there wasn't enough biology underlying, or understanding of the biology underlying pruritis to have more rational drugs. I do think we'll be entering an era where that is available. Not quite there, but we're getting there.
I am concerned, to my point about, the discipline of the field and the role and credibility of patients, that certainly we have a significant role and interest in ensuring that there is robust investment in liver drug development. I do think that there is also a responsibility to give realistic advice about how those drugs are introduced to market. And so in NASH, certainly, I think that people are being responsible for recognizing that this is, when a drug is approved, it will be likely in a polypharmacy environment. There will likely be, multiple mechanisms of action, multiple NASH drugs, and then multiple drugs for the patient's other concurrent conditions. And so, they should, as my pastor would say, govern themselves accordingly when it comes to price. Mm-hmm. What I would hope that as we see greater development in pruritis and other symptomatic and hopefully real resolution of disease in for other liver diseases, that they recognize that from a patient advocacy perspective, we're not going to support anything at any price, and that we do feel a responsibility to the field as a whole, both for the investment and for the access and to the future access. And so some of the pricing that's come out about pruritis, the math does not work for patient advocacy. And so, we have concerns. And again, if there had been conversations, as there were in NASH, with patient advocacy early enough, there would not be, a risk of backlash at this point, post-market, for something that patients certainly want, more resolution for pruritis. And so I'd ask folks not to have unforced errors and create problems when there didn't need to be one, and to have very credible strategies. And, Roger, your background is in, market modeling, and I think they don't often or model for effective patient advocacy or pressures back on how access is defined. And so I would recommend that.
Well, it's telling how much modeling comes out of the finance departments, right? Because we know, number one, and number two is the reality is that it's a misnomer that people respond to their market, that companies respond to their markets, companies respond to where they get their money, which means if you're headed into the market for the first time, you're going to go where your investors want you to go. And by the way, until you've got your $10 billion, until you've got Lipitor, or you've got Keytruda, or Humira, you're going to go where your investors tell you to go, and then you wind up with prices that are just too high. Right. And there isn't enough patient sensitivity anywhere in that system. I think that's definitely true. One last question, okay? We're back here in a year. We're having the same conversation. What's the one thing you think will have changed, and what's the one thing you hope will have changed?
Well, maybe I'll jump in first. What I hope will have changed is to see the first successful or approvable trial. We shouldn't forget that obeticholic acid was successful by any measure. Whether it's suitable for FDA approval, I think, remains to be established. But I would like to hope to see the next drug, presumably that'll be Madrigal's drug, showing some signs that we're starting to get over the starting line. Not the finish line. And what I expect to see is continued progress in the non-invasives and the more sophisticated analyses of tissue, because those can be done with existing resources. Now, I could say I would expect to see a successful new drug, but we know that that's a 3 to 5 year process from when the clinical trial starts. So I don't think in a year we're going to see a breakthrough beyond what we hope will happen with the Madrigal drug. But certainly on the technology development side, using existing reagents, tissues, sera, I do expect to see a lot of progress there.
Okay.
I both think and hope that in the next few months we will have, both from the patient advocacy side and the researcher and the drug development side, have strengthened our relationship with FDA to the point where there is clarity about the mark that we need to meet, and a commitment to move forward once we have met that agreed upon mark. And the second thing is I foresee that we will have reached a level of precision in which non-invasive technology to use for what purpose, that we can have meaningful conversations with primary care and clinical management will be much easier.
So I'm going to give a different answer than I would have given before the first session this morning, because we didn't get to this because everybody had to run out of the session. But on the way out the door, Mazen made the comment that if he were advising investors, he would tell them to invest in 22 heavily, because he's pretty confident that in 23, maybe even in 22, FDA will sign on to digital histopathology. And if they do that in the next year, that's a huge difference.
Right, and that's exactly what I'm saying.
So that's the one I expect to see.
Yeah.
The one I hope to see, so when I came here for the first time in 2019, all anybody wanted to figure out was how do you get fibrosis down one level. And I think one of the emergent realities is that for an increasingly large proportion of patients, if you can stabilize the liver, that's a good enough outcome. Right. If that becomes accepted widely enough, we can find ourselves in a situation where people will start walking themselves into things like SEMA and TERSA when it shows up, which clearly have the ability to drop weight, do all the right things, and stabilize the liver. If we're willing to accept that as a goal, I think we can move faster even without approvals.
Absolutely, and we've known all along that the FDA considers preventing progression from non-cirrhosis to cirrhosis a hard endpoint. That's not a subpart H, because everybody, FDA included, understands that cirrhosis equals complications, cancer, potential death, or transplant. Non-cirrhosis avoids almost all of those, with the exception being that occasionally cancers can arise before patients are fully cirrhotic.
Yes, and my point, I think, is that, and I agree with that, is that, and they know that, in the last 6 months, the volume around stabilizing is enough has gone from about here to about here. If it gets about that much higher, people are going to start taking action on it. And to me, that's the single greatest hope for how we help the most patients fastest.
Couldn't agree more.
I absolutely agree, and I'll help do all I can to raise the level on that. I'm not trained in medicine, but I am trained in logic, and so stopping progression of the disease has always made sense to me and to our patients. Helping to share that with others is a fantastic priority.
Well, this has been a great conversation, and thanks so much for each of you knowing that you couldn't make it for the first round, saying, hey, I'd like to come back and do the second round so that we got to do this. And I can't imagine two people I had more fun doing it with. So
that's very kind.
Thanks
Always
both
enjoy
of you.
it. Count on me.
And I will,
you won't get rid of me so fast, Mr. Green.
I'm not looking for, look, you know, two Jewish guys from Long Island of roughly the same age. You know, if I get rid of you, then who's left?
That's what we used to say, you're a Landsman.
Yeah, we are Landsman. It's true. It's true. Okay. Thanks, everybody.
And now let's join Louise and Roger in sharing some reflections on the Friday program, including impact of diet on clinical trials, use of older medications to support patients with cirrhosis, the challenges small companies face when bringing a drug to market, and the increasing need for accumulated data to answer the big questions facing Nashville today.
Louise, let's stick around for talk for a few minutes about how to wrap up what will be certainly an unusual episode by our standards. First of all, personally, the point that Michael made in his talk about the small companies doing all the development, I think the importance is undervalued. Although Stephen, I think, did it on that one issue. But if you listen to Vincent Wong's presentation of the aldafermin data, he did, I think, as good a job as anyone could have done as explaining this was a commercial problem. This was not a medical problem. The medical problem isn't the drug doesn't work. The medical problem is it's injectable daily. And that's a tough row for anybody to hoe. As you know, as GLP-1's proved, if you can get it out to weekly, it's pretty good. Daily, that's a hard thing to do. But beyond that, all these other places that come into play in terms of people don't have the time or the budgets to do the big studies that would protect them against quirky outcomes.
It sounded very much like that. The guys this morning did a really good summary of the day and actually how exciting the day was. I think Michael was right at the beginning. We're all looking for the big drug that's going to cross the market and give the income. That's hard to achieve. One thing that you and I texted about was Michael's comment about the underrepresentation of diets and lifestyle in these trials, and they can completely skew a trial. If you don't know how many vegans, how many veggie, ovo, and lacto vegans and vegetarians and things like that, they particularly skew your data because their BMIs are lower, they've got an energy intake that's lower. And being a vegan, being a vegetarian, as we all know, does not mean that you're particularly healthy. It's not necessarily a healthy diet because you substitute other things into it, but it can be. So all of those come into play. What really excited me from hearing the summation this morning is the ability to look at combination therapies, the ability to look at fast-acting medication medication that can bring you into a stable position whereby you can add in some of the other medications. And that for me excites purely because we've seen it in the world of hepatitis C. We started with daily or tri-weekly, to be fair, injections of interferon. It didn't last very long. We pegulated it. We went to weekly. It became more tolerable. But it was also about that coaching through that intolerability for many to get them through. So where the discussion went yesterday was, yes, any form of subcut injection that's daily is a barrier. We see diabetes not be a barrier for insulin, for example, where you need it. But then moving to AI and that pathology and the histoindex, it is our obligation as scientists to use the samples, particularly histopathology samples, in the best way that we can. And getting the most accurate reading is obviously going to be by AI. It picks up so much more. It can see things. We are constrained, as Quentin said, by thinking about within the human brain and the way that a histopathologist and therefore machines need to see what we see. No, they don't. They see way much more. So removing our barrier to thinking allows AI to really tell us what is there, not what we preclude. That was the excitement for me yesterday. It moved through the whole continuum, and they've summarised that this morning. What we didn't touch on this morning was the presentation on the cirrhosis session that Manal was leading and that I think Vijay presented on, and simvastatin in the clinical situation, reducing and improving some of the outcomes which people weren't aware of before. And his cohort was all cirrhosis. It wasn't just NASH. It wasn't just
it's the cirrhosis network. It's all cirrhosis.
Yeah.
But look, Manal's only come on the podcast 3 times on this in the last 6 months. And by the way, has gotten really large download audiences for the twin ideas. The stabilization is an adequate goal at that point in time, and you can do it with older drugs, notably statins.
Absolutely.
People are actually downloading that fairly frequently.
Well, if I want to stabilise my patient as a physician, and which they do for the next drugs coming, you need to maximise what we've got currently. And what we learned certainly on when she did that on the session that we did with her, she put out a load of suggestions to help improve management. They're not going to work for some, but they will work for others. That was an important session. We haven't had time to cover everything because we'd be doing the whole day again, but I think the guys this morning just summarised the breadth of what came out, which is why I was on the science yesterday.
I had an optimistic reaction and a more, I'm going to say skeptical, not pessimistic reaction. The optimistic reaction is all the stuff we're talking about. And specifically, the idea that when I started working in this space 3 years ago, all anybody wanted to figure out was how do you get fibrosis down levels so you can get a drug approved. The science has become so infinitely richer. And I think part of that is because OCA didn't get approved, that people weren't shooting for a target that goes, how do I supplement OCA? It's what do I need to know here to get this right? And that's been fantastic. The thing that worries me, and part of this is my comment about small drug companies doing development work, is when I was consulting companies on commercialization for marketing research that we did, the point I used to make is, it's not very hard to walk 100 yards unless the 100 yards is from one bank of a river to the other bank and the river is deep and it's got currents. And then unless you're above, my co-religionists don't believe anybody ever walked on water. So that would be the end of that one, right? And maybe it turns out that the bridge is 10 miles down the road. So now you've got to navigate your way 10 miles down the road, bridge 10 miles back up to walk 100 yards. There are so many moving parts in this, and it's so challenging to see the whole elephant as compared to the 6 blind people touching the elephant, each know what they touch. It's so hard to visualize the whole elephant. When you get outside people in this room and the folks who are really the top tier academics, as you start going into more commercial spaces and more finance spaces, it gets harder to do. So I start to wonder, how do you actually get to where you have to get to? How do you go one step at a time and plot that one out? I think it schools out yet on whether that's going to be easy to do, hard to do, or impossible to do. But it does give some cause for pause. When I listened to people yesterday talking about what they took out of the FGF21 talk was, pegfilgrastim fell off the road in 48 weeks, and adiponectin and what's going on here, and can you really trust anything until you've got 48-week studies? And Stephen's point was right. There are other reasons that might not be a 48-week drug, but if you give it an F3 or an F4 for 24 weeks.
But I think,
go ahead.
I think when we did the session where we discussed the FGFs, didn't he comment that lots of people have different FGFs and our own makeup says what we'll do? And I asked the question of whether we can phenotype people for FGF, to which he said that's not been done yet.
Right.
And pegfilgrastim may well just have failed and been a spectacular failure because it was the wrong phenotypes. And that's where precision medicine, and we touch on that a little bit, comes in. Now going back to that study and seeing if you can really phenotype them for which FGF they're going to respond to, that's where it gets exciting. I know earlier on I talked about basically precision medicine is in the individualization of the medicine for that person from the minute they walk through the door, rather than just the expensive end at the other side. But when we're talking about drugs that with billions invested in them, this is where you are right. Small companies without that finance can't do that work. And therefore, are you on a hiding to nothing? So the maximization of precision medicine in clinical trials with a consortium, with the opportunity to combine their data, as I've discussed before and Stephen mentioned today, we need to come to this together to solve the problem, to be able to get the drugs to market. I don't think we're going to be doing this as individual spots on a Dalmatian. It's the Dalmatian at the whole that's going to get through it.
Yeah, that's exactly right. And by the way, I've never seen a spot on a Dalmatian that won a dog show, but I have seen Dalmatians that won dog shows. I totally agree with that. And in that regard, maybe the most encouraging thing that I heard yesterday wasn't even in the meeting per se, but it's all the organization that's going around consortium to compile all the data. There are tons of data at this point. We're thick with data and short on integrated insight. And I think Nimble and Litmus are starting to prove that, and what Quentin said this morning that was interesting is, all right, so Nimble went after it one way, Litmus went after it another way. When you take the two insights and you combine them and you don't ask who's right and who's wrong, you assume they both did good work. So how do we take everything we learned and put it together in an integrated package that people can execute on? I'm going to go back to your precision medicine comment on that, which is that there isn't going to be enough resourcing to do what these folks think of as precision medicine every time somebody walks into primary care. So it might not be a bad idea to look for a different nomenclature for what you're describing. Because precision medicine, what happens is, things become their own brands, right? So precision medicine now has conferred on it expensive testing, very, very sophisticated understanding, because precision is that kind of thing. If you go back to Stephen's metaphor about the Wright brothers and fifth-generation strike fighters, that's about increasingly precise flying over time. So I think your point is right. I was watching their faces, which you couldn't see. It gets lost a little bit in the metaphor when you use that term. So the question becomes, how do we start to thoughtfully, for the first time we see a patient, understand what is within the ken of the primary care doc to do? And how can we help the primary care doc get information efficiently that will help them do that? If you go back to Alina's comment at the end of the year about using AI to go through medical records, that is precision medicine in a sense. It's a lot closer to what you're talking about today, but it wouldn't be defined as precision medicine. It's going to get defined differently somehow.
Oh, absolutely. They do it. For example, for HIV, they look through the alternate diagnosis, herpes, and other things that could alert you to that person being at HIV risk. So they go through with AI for a lot of stuff. So Lena's absolutely right. We need to be using all of these. But then saying that you've got to have medical records that are uploaded, that takes the cost of some of them. Those things could be prohibitive as they're seen at the moment. But I think in the future, with electronic records, more or less, certainly in the Western and developed world, being fairly standardized, it's an opportunity.
Agreed.
And I'm looking forward to today. That's all I can say.
Welcome to today's business section.
I want to keep this short. Basic headline is, oh my gosh, are a lot of you listening to this podcast?
Remember way back in September, when getting 3,000 downloads in a month sounded like a dream?
Yeah, I remember when 3,000 a month would have made us really happy. Getting us 4,400 downloads in October made us virtually delirious. And following that up with 4,400 in November and 4,200 in December made us crazy giddy, but nothing prepared us for this month. As I mentioned last week, we had roughly 2,300 downloads in the first 10 days of the month, or 230 a day. We're slightly off that in the last 9 days, but still averaging close to 200 downloads per day. So the punchline is that at around 1 p.m. this afternoon, January 19th, we crossed the 4,000 download for the month plateau, a level we'd never hit before October and never hit before right at the end of the month. 5,000 for January looks close to a lock. 6,000 isn't out of the question. These numbers are just crazy big, but they're also exciting and really gratifying. Thanks for sticking with us. Thanks for downloading. Thanks for being part of this.
What's coming up in the next 2 weeks?
The next 2 weeks, we'll see 2 new episode concepts. Next week, Jeff Lazarus, Jorn Schattenberg, and 1 or 2 other guests to be named will join us to discuss today's webinar kicking off the Innovations in NAFLD Care 2022 series, a offshoot of the Wilton Park work and the advocacy for public health agenda that Jeff talked about with Jorn and us in December. This program is one piece of the emerging global effort to build a multidisciplinary clinical care pathway for NASH diagnosis, which also brings in some of Ken Kusi's work that we talked about last year. Treatment, monitoring, whole 9 yards. SurfingNASH is a media partner for this series, whose next event is a hybrid meeting in Barcelona early in May. I'll talk more about that as we get closer to it. That's this week. The one after that is in part an answer to the most common question I get when amongst fatty liver people, who always ask me what Louise is really like. Well, you'll find out on February 2nd when we post the podcast Louise, not me. Louise is hosting on January 31st while I'm on holiday, focusing on the roles that nurse practitioners, nurses, and other affiliated health professionals play in the supportive care of fatty fatty liver patients. Louise is pulling in some fantastic guests from her own past experience. The topic is fascinating. I think it's going to be a great time.
The vault is open.
We already covered the idea that lots of you are listening and downloading. The question is, to what exactly? So here's my answer. First, the Parish-NASH Imaging Conversation 46.2 gained another almost 100 new downloads in the past week to go to 1292 as of 10 p.m. Eastern Time Monday night, 1298 as I'm talking to you right now. Conversation 57.1 from the AASLD wrap-up episode continues to do well, moving up to third on the all-time list. And further back in the vault, take a listen to last year's NASH-TAG episodes and this past December's AASLD sessions. They're all doing really well. I want to end this business report by thanking our team, the magical Mike Wilson, the creative and highly energizing Eric Rounds, the Steve Ennin, who keeps knocking it out of the park every week as we build strategy. So let me just say this. We all believe that if you folks didn't get so much out of listening, I don't know where we would find the energy to keep doing it. So thanks for the energy, and thanks for being the wave that floats us. Beyond that, Omicron continues to put a dent in all of our lives. I've been exposed to at least one person close to me who tested positive for Omicron every 2 to 3 days since the new year. That's 3 weeks ago, folks. I keep testing negative. However you're testing, and whatever else besides COVID is testing you, be patient and show good health judgment as we go through one more round of this mess. Stay safe, mask up, surf on. Look forward to seeing you all on podcast next week. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com and we will answer on the podcast or the website.